ahead and introduce our speaker for today. Today we have Dr. Anthony Williams. He's joining us from the EPA and will be giving us a very interesting talk about the chemical dashboards, an integrative data hub for environmental science. And with that, I will like to give you the floor. Go ahead and get started.
Okay, thank you. Can you hear me okay?
Mm-hmm.
Okay, great. Thanks for the opportunity to present to you. I greatly appreciate everybody taking on yet one more opportunity to sit in front of a screen and listen to yet one more online webinar, because I'm sure we're all doing a pile of them. So hopefully this is interesting to you. I have a lot of slides. I was hoping to show you a live demo, but to be honest, EPA is suffering some fairly significant network issues for the past 48 hours, and things are a little fragile at the moment, but I'll do my best. If we have time, I will go into a live demo. So I get to give the presentation. However, I am standing on the shoulders of a lot of people. So the data I'm going to talk about has been in development for over 20 years. We have a lot of chemistry data that we have been harvesting and curating, and tens of people have been involved in that. We have a lot of database people that have come and gone. We have informatics people that have come and gone. The application I'm going to show you, while it's been online for just over 5 years, is sitting on the rich efforts of many people. So that's why I list an enormous cast of characters. I really couldn't do justice to all the people involved. The website I'm talking about is publicly accessible. So by that I mean anybody in the world can use it. You don't have to register. You don't have to log in. There's no boundaries or limitations to how you use it. It is not an open source application in terms of we don't yet have it available for people to download all of the code. The data you can take away and use to your heart's content. I'll talk more about that as we go. Ultimately, our intention would be to make this available for people to run inside their institutions, but we've been so busy building this over the years that we're not there yet. So open for sure, as fair as we can make it for the moment, but not open source. I'm not sure whether these things that are blocking up keep hindering the slides, but I'll keep going. So it's 880,000 chemicals that you have access to for the moment. Substances, not just structures. We have a lot of experimental and predicted phys-chem data, hazard data. We are trying our best to build an application that can be used by different types of people. So to support risk assessment, to support informatics needs for chem informaticians, to support people interested in toxicity, people who are trying to do chemical structure identification for mass spectrometry. So this application can be used in many ways. I was one of the original founders of ChemSpider. So if you've used that, that was also built so that many people could use it in various ways. So this is a flexible application. We have been measuring bioactivity data for over a decade. Tens of millions of dollars have been invested in that. So those data are available. We have efforts that have been led for a number of years now to harvest information about chemical substances contained within consumer products. We never go it alone. There are so many efforts online that people are working so hard to bring up their own applications, make their own data available, that we also do our best to link to those resources so that people can come through our link farm, as it were, and migrate out to these other applications. And that includes our own agency, other government agencies, and of course, tens of public data resources. We have built an application that integrates directly to PubMed, so that allows people to come through our application and use our abstract sifter, as we call it. And we've also built a generalized read across application inside the dashboard that can be used. I'm not going to have time to be exhaustive in everything I show you today. There's simply too much. A hands-on demo today where people are working along with me is about 2 hours minimum. We're not just working on singleton chemicals where people can search for a single chemical at a time. That is a limitation for the ChemSpider application I was involved with. but we allow you to do batch searching for thousands of chemicals. So that's a general overview. And to quickly walk you through what we have at the moment, it's a simple search interface that uses alphanumeric text. So CAS numbers, synonyms, identifiers. We don't yet have the structure substructure similarity search built into the application, but we do have a prototype available that we will be integrating. Gives you access to the tox data, the bioactivity data. We have some basic informatics plugged in terms of doing similarity search for the moment. As I said, we have structure and substructure built as prototypes. This is the read across module. We won't touch on that in much detail. This is the PubMed abstract sifter and the batch search. By the way, all of these slides will be circulated. I will send them on later, and I will also put them on my SlideShare account for you to download if you need them. So that everything I'm showing you will have access to. Let's go just for the chemical search for the moment. If I want to search for bisphenol A, it's a type ahead search. I can find all related chemicals that contain the bisphenol string. It is a type ahead search. If I want to find everything according to a substring, there is a checkbox to allow you to do that, so you can search on the substring. Here we're landing at the bisphenol A page. This is the chemical details page. All of the, let me switch to pointer here.
Dr. Williams, I believe we can't hear you.
Can you hear me now?
Yeah, we can hear you now.
Okay. So, yeah, sorry, there's a button on my headset I accidentally hit. So some people don't trust Wikipedia content. I worked on Wikipedia for 3 years. I looked at 8,000 chemicals, and a group of us have worked very hard to elevate Wikipedia content in terms of data quality. But I understand some people don't like it. It's hard to make everybody happy, but we find this a useful snippet. Intrinsic properties, of course, every structure that we register, we have an underlying chemical registration system called ChemReg, which is built in-house. This allows us to register all of our substances with CAS numbers, identifiers, mappings to related substances like metabolites, degradants, etc. So that chemical registration system is what serves this application. The application sits on 8 separate databases at the moment. For every structure that we register, of course, we have formulae, mass, monoisotopic mass. This enables us to do the advanced search capability here to support our colleagues in the agency who are doing mass spectrometry, non-targeted analysis, etc. Structural identifiers will be a link into the UPAC name. It will give you INCHI strings, INCHI keys, smiles, etc. And as we go through here, there's linked substances. These are chemicals that would be linked as salts, multi-component chemicals, presence in lists. You'll understand lists by the time I'm done. We have about 270 of those linked in. And then quality control notes is all related to our curation efforts, and record information tells you what level of quality there is for a particular substance. We have curators, 5 curators full-time who are looking at the data every single day. We're adding new data and into the underlying database. And we have 5 levels of quality that we have there. I'll explain that a little more as we go. And we also have 2 additional levels of quality where we are not yet finished curating the data. So while we have access to it internally, we do not release it to the public. On the left-hand side, we have the navigation subtabs, and this is where you will dive into additional data about the particular chemical. And this is where we will spend some of our time navigating through during the presentation. So this is showing you diving into the physical chemical properties. So you can see here for bisphenol A, for this small list of properties here, we have certain experimental data available. And where we don't have experimental data, vapor pressure, flash points, etc., we can predict those properties. We have built QSAR models based on thousands, if not tens of thousands of data points that we have harvested and curated over the years. So we predict those when we don't have the data. This is showing you the experimental data that we have available for the chemical. You can actually see the links out to specific journal articles with the value. hover over any of these and it will give you that direct link out. And this is the predictions. So we don't favor one prediction over another. There are multiple QSAR predictions out there. Some of them are commercial. For example, ACD Labs is a commercial entity that would sometimes puts predictions in there. We have our Opera models. I will tell you more about those. EPI Suite is freely downloadable. It's been built up over almost 2 decades. And is one of the prediction suites that we have available from EPA. But there are differences in what we can show, because here you can see we can't tell you the calculation details for certain predictions. We don't have those calculation details. They're not supplied to us in a manner that we can expose. But for certain of the QSAM models we work with, we do make those data available. This is an example now where we, for this particular chemical, we have clicked through to see the Opera model report. The green visual cue is telling you that it's inside the applicability domain. If you're colorblind, red-green colorblind, then the inside AD is inside the applicability domain. This is showing you for log P. This is the particular property. This is the distribution of the training versus test set of the log P values, log K or W, some people would call it, that have been used to build the QSAM model. And this is a 75-25 split training versus test set, and the cross-validation efforts. So these are visual depictions of the amount of data that we have available, and these are the performance of this particular algorithm to give you a predicted value for log P. One of the things I should point out is that right here you have the QMRF. This is the QSAM model report format. Details that tells you exactly how the models were built. It's a PDF file, you can download it, and it will explain to you what descriptors were used from the PADEL descriptor data set. It will also tell you what software we used to build this, and in theory, you could repeat exactly what we've done with our training set and test set. All data are open, and you will be able to rebuild these models should you choose. Our data are open. You can download them, take them, mesh them in with your own efforts, and rebuild the models yourself if you wish. Opera predictions are available from GitHub. You can download all of the code involved with Opera. You can run it locally. And since the models were built with a gentleman called Kamel Mansouri, he has continued to extend that suite of models. So there are even more predictions available than we are just showing on the dashboard. We have access to exposure data for chemicals. So we like to know what is a chemical used for? What products is a chemical in? At what chemical weight fraction are those products available? Also, monitoring data tells you when those chemicals are being monitored through NHANES. We also have predictions based on some of our models that are being developed inside the center. And where available, we show you production volume of these chemicals. So it's one application that is giving you access to all of these data. Of course, the data are incomplete. It's always changing, and we're doing releases about every 6 months to try and stay on top of that. We do a lot of work to pull together synonyms. So this is helpful for when you're doing the type ahead search, or you're using a batch search and you have a list of chemical names. If we have an incomplete list, we're not going to be able to do that matching for you. So we try to pull together as many of the appropriate synonyms, identifiers, CAS numbers, Barstein IDs as possible. There is a distinction here between the CAS numbers that we host, where this is active, and there can be multiple deleted CAS numbers also. So for bisphenol A, if you scroll to the bottom of the page, you're going to find there are 5 additional deleted CAS numbers. The community remains rather confused around CAS numbers, I find. People think there's one CAS number per chemical. The reality of it is that we have examples on here where there can be 300 deleted CAS numbers against one chemical. So one active CAS up to hundreds of deleted CAS numbers. We have to have them all so that when you're doing these searches, we surface the chemicals correctly. This is the link access I was telling you about. So this is the link farm directly here. So for bisphenol A, Where possible, we have mapped up to external resources so that you can find information about the chemicals with a single click. So for BPA, you want to find it on PubChem. This is a direct link. The way we do this, it differs from site to site. So for PubChem, for example, we have registered our substance list on PubChem, so you can find it there. You can download it with PubChem IDs. And because we've done that, we are able to harvest back the PubChem IDs on our side. We register them directly into our database, and that gives us a link. Same thing happened with ChemSpider. However, we have not been able to do that with every resource because many of the sites will not allow you to download the data sets and completely. So we do things like on the fly, cast number searches on the fly, Inchi key searches on the fly, smiles. So if we're going to do predictions of NMR spectra, for example, here, we're pushing out smile strings. Those smiles, of course, are consumed by the prediction algorithms. In terms of mapping out to external resources, for example, Mona or MZ Cloud, we're either using a combination of cast number and Inchi key or something of that nature. So it really depends what the sites are that how we've mapped to them. This is an example of I know there's people on this talk who are going to be very interested in the mass spec linkages. So we have linked up to a number of sites to enable us to dive directly into things like infrared spectra on PubChem, NIST spectra, MZ Cloud, Mona, of course, and MassBank. So these linkages are based, as I said, on a list of various identifiers depending on what we can use at each of the sites. This is an example of going in to the NIST webbook using a cast number for the linking purpose. So this is on cholesterol. This is going on to, of course, MassBank of North America. So we can dive directly into that. So we feel this linkage is very important for people to understand the hub that we are for all of these data externally. We're trying to help people navigate to resources as fast as possible. We can add these resources quite quickly. So in about 6-7 months ago, I was giving a presentation. There was a gentleman in the room who followed up with me on a phone call, and he said, you know, do you have Antoine constants available for chemicals? And I hate to plead my naivety to this audience, but I didn't know what an Antoine constant is. Maybe at some point in my life I did, but at that point I didn't. And I said, where would I find the data? He helped me find it on the NIST webbook, and we linked up Antoine constants within about 20 minutes of him pointing us to it. So we add to this set of external resource links on a regular basis. There's a whole lot more data that I can go into there. So the hazard subtab will take you to the toxicity data that we've been harvesting over the years. So there are 56,000 chemical substances with over 700,000 toxicity data points extracted from 30 public toxicity resources. We curate these data as best as we can. They're going to include NOELs, low ELs, cancer slope factors, unit risk, et cetera, et cetera. So all of these data are brought together, homogenized, and made available through the hazard system. The bioactivity data is the ToxCast and Tox21 data that we have available. So that's going to be over 8,000 chemicals have been measured in up to 2,000 individual assay endpoints. So this is in vitro bioactivity. So we're probing multiple different targets. We have the dose response curves directly available. We have, and this can all be downloaded if you wish to get access to all of our in vitro bioactivity data that we've measured over the past decade. You can download it in a SQL database, install it in your own systems, and make it available. We also have real-time predictions that we can perform on the website, et cetera, et cetera. So there's a lot more data in there. And out of this presentation, which I'm thinking of as a general overview, if anybody would like to have a webinar where I could give them online training and allow you to ask questions as we go, more than willing to do that. Just follow up with me afterwards. But as I said, we have some challenges with the network today, so it's going to be a little tougher. So I was talking there about bisphenol A, and singleton chemicals, very useful. If all you care about is one particular chemical, you want to find out what we've got available, then this is the path to go. Input the cast number, the name, deleted cast number, Inchi key, etc. However, most of the work that we've been focusing on is because we've been harvesting these data and integrating them together to put them onto this website. Most people are interested in batch searching. So, for example, if you were interested in hydraulic fracturing and you wanted to take a list of chemicals associated with fracking, come to our site and find all the tox data. Have we run in vitro bioactivity screens on any of the data? What are the phys-chem properties? Log KOW, water solubility, vapor pressure. You want to harvest all of this as fast as possible, pull it together. We've done the heavy lifting for you by integrating this from eight databases into a single dashboard, and then layered on top of it a batch search. So as an example, I was at a meeting in Pennsylvania and Philadelphia a couple of years ago. It was a mass spec presentation. The presentation was regarding opioids. And my recollection was it was Marina, the first author here, who's giving the presentation, I think. And she was talking about how much work they'd done in order to pull together a list of opioids just to get the formulae in the masses. And that is valid and appropriate work. You might be using PubChem, you might be using any of the lookup services that are available to try and do that. One of the challenges is data quality, I'm afraid, for a number of the resources. We're all struggling with data quality. We work hard on it. But our curators have been able to pull together in a very highly curated system a lot of the data that might be of interest. So the question, this is the presentation she was talking about this particular work in her paper. While she was talking, she mentioned this. I immediately downloaded the paper. I went into the PDF file. I harvested the list of names directly out of the PDF. It took me longer to harvest the names out of the PDF appropriately than it did to do the following, which was go into the batch search, select here the chemical name, because these are names. If there were CAS numbers, we could mix CAS numbers in INCHI keys and just throw them in here and hit search. And then immediately it will tell you that we have approved names. So these are the primary preferred names on the chemical, and this is a synonym. And here's a list of identifiers. These identifiers are our substance identifiers. They are unique to us. They are already in Wikipedia. They're connected into PubChem. They're connected into the Chembl resource, the Unichem resource out of EBI. So these substance identifiers are already proliferating on the internet and connecting a lot of the websites together. So that's great. Okay, now I have a list of identifiers. Big whoopee. What can we do now? Well, as I said, we can add lots more information. So on the batch search, you have the opportunity to add more in. So I don't just want to get the identifier and the name that's associated, but give me the CAS number if it's registered. Give me the INCHI key. Give me the UPAC name. Whatever I check here, and the list is much longer than this, then pull it together and allow me to download that data set. That data set can be downloaded in a CSV format, in an Excel file format, or even an SDF file format. For those of you who are doing chem informatics, you might choose to download it with all of the structure layouts already embedded in it. So you can see here CAS number, formula, monoisotopic mass, MS-ready smiles. I'll mention MS-ready smiles shortly, and then on and on and on. As you go across the Excel spreadsheet, everything you've selected is embedded. This can include all of the opera predictions. So the 13 different endpoints that we've built models around, you'll be able to get those predictions. Notice here, we did not have something with the name naltrabin. It turned out that we did actually have the chemical structure, but we didn't have the synonym. Sometimes this can happen because of a spelling error. In this case, this is the correct spelling. We just didn't have the synonym. So added that to ChemReg, and away we go. We were able to harvest the data. So this is useful and used by many people. Now, I showed you the searching based on chemical names, but to clarify, the batch searching can come with other search capabilities. So you can search based on mass, formula, MS-ready formula. You'll hear MS-ready shortly, INCHI skeleton, etc. So there are other ways to do this batch searching. One of the common questions is going to be, do we have web services to allow you to access the data? I'll talk a little more about the API and the web services later. So 882,000 substances has been built up over the years, over 20 years, as I said, and a number of lists have had to be registered in order to build out this substance list. So we add a number of these chemical lists to support specific efforts of the agency. So for example, actually this number top left is wrong. I think we're at 273 now. So the list of chemicals will change depending on our activities. So right now we're very interested in PFAS chemicals, perfluoro, polyfluoroalkyl substances. When we were working on hydraulic fracturing, we would work very hard to curate those lists. So from the top of the application, you can go to lists of chemicals, and this will bring you a set of lists. Here I filtered it based on mass, so you can find the mass spec DB here. This is a publication that was put out with Emma Schmansky. This is MassBank EU, mycotoxins from MassBank EU. So this is a subset of those lists. Here's one example of something we were working on last year. So we had a lot of interest in the volatilome. So this is a subset of compounds detected in human breath. And the reason for doing this type of thing is if, depending on your area of investigation, you might not care about any of our 882,000 chemicals, except for the ones that have been detected in human breath. And one of our colleagues, now retired, Yoke Pleel, was specifically interested in chemicals in human breath. So this was a list that we put up. We show you the linkage to the journal articles, etc. We also threw one up for the saliva subset. This is an example of where our interest in PFAS is really focused. So I believe we have the largest single collection of PFAS chemicals on a database today that are at least curated. It's not difficult to go find larger collections of PFAS chemicals. The challenge is that many of these PFAS chemicals are being done by people using name to structure algorithms. So for example, they will do text mining, they will take a PFAS name, they will convert it using one of the commercial tools or something like the OPSIN system, which is an open source name to structure conversion algorithms. They will take the outcome of that and they will throw that up into a database. No curation, no efforts. Well, I've looked at a lot of those data sets, and a lot of it is being done from patents, and a lot of the data in patents, unfortunately, are name substrings. Many of these chemicals turn out to be radicals. They're fragments of chemicals. They're not real. So we're trying to do this in a much more focused and curated fashion. So these are some of the lists that we have available. These sets of chemicals are ones that we're studying directly through in vitro bioactivity right now, these two right here. We have purchased over 400 PFAS chemicals. We are pushing them through bioactivity studies. We are measuring property data, et cetera, et cetera. We also have the list from the Swedish Chemical Agency report. We harvest this list out. There's a lot of work done by Stellan Fischer and his group in the Swedish Chemicals Agency. But then we go through, we harvest the data, harvest the collection, we curate it, we map it to CAS numbers, et cetera, et cetera. So a lot of work going on this set. We were approaching 7,000 structures, and I want to talk about where our focused efforts. So we had about 7,000 structures 6 months ago. When we last did our release, it had jumped to over 9,000. Why? Because we are absolutely working on this effort. This is high-profile effort for our agency to gather more chemistry regarding these chemicals. This includes going through literature articles, our own analytical laboratories are identifying new chemicals in the environment. So we are pulling these data together. And I mentioned that we have the largest curated collection. If you compare it with PubChem, in terms of this particular substructure right here, PubChem will give you 220,000 PFAS chemicals, which is an incredible jump considering we only have less than 10,000. But again, the reason is most of this is coming through this Shaw Campbell effort, which is through EBI. They're pulling the data from patent authorities. And whether I look at Google patents or whether I look at Shaw Campbell, I find a lot of these chemicals just simply not correct. So That doesn't mean they're not all correct, just many of them are not. So we're doing this curated effort. Going back now to the interface where I was showing the fact that we have these intrinsic properties for a chemical now, because we have simple linkages already built into the application. If you have a formula, this chemical structure, of course, has a formula generated at registration, then we can click on one button, find all chemicals. For many of the people on this call, this should be second nature. Believe me, there are people who do not know that this is this simple to do, that you can go into some of these informatics tools and do this. So we're trying to make it as easy as possible to do it. So from here, PFAS, here's all of the variants of PFAS that we have available, where you've got both the linear form and the branched form. When people talk about PFAS, perfluorooctane sulfonic acid, they miss the fact that it can be both linear and branched. In general, PFAS itself means the linear form. It doesn't have to be. And then we run into even more complexity because people believe that PFAS is any salt form. And of course, different salt forms have different CAS numbers map to different data. Things get very complex. We're trying to deal with all of that with our curation approaches. Another list that we built up is the disinfection byproducts list. So we've been working with Susan Richardson in South Carolina. She has been writing reviews on DBPs, disinfection byproducts, for a number of years. This particular one goes all the way back to 2003. Her last one was in 2019. And what we're doing is we're working with Susan to make sure that we put the list up. The version that we had initially was 887 chemicals in the disinfection byproducts list. Having worked with Susan last year, we're now over 600. So we are continually updating these lists. So if you wanted the list of all chemicals identified as disinfection byproducts, I say all as referenced through her review work, and she's very thorough, you could download that list directly. An example of the mycotoxins list from MassSpecDB. Just talk about complexity of structures and how hard we work to curate data for a moment. So this is vomitoxin, not one of the simplest chemicals you're going to have to draw because of the complexity of stereochemistry here, but it is an example of complexity in chemical structures that gets thoroughly broken in public domain databases and people not understanding just the most simple detail of how to represent a chemical structure, such as the narrow end of the wedge points to the stereocenter. As an example of that, I was in an argument on Wikipedia curating one chemical structure for one stereocenter for 3 days, trying to figure out why the person that was engaged with me, I say arguing, it was collaborative. We were trying to get to consensus on how to represent a particular chemical. The chemical was tacrolimus. If anyone cares, go to Wikipedia. You can go into the discussion and you can see how we talked about this structural representation. And it took me 3 days before I finally communicated to the person, were you ever taught that the narrow end of the wedge points to the stereocenter? And he didn't know that. And as soon as he got that, then we were able to agree on how a chemical gets represented. So vomitoxin has got multiple stereocenters here. As you can see in the structural identifiers, we have the UPAC name, generated algorithmically, fraction of a second, commercial software tools from ACD Labs. The smiles, of course, differs on what toolkit you use. The INCHI string, this is the standard INCHI and also the standard INCHI key. So with that INCHI key now, one of the things that we like to do is you can see below that we have this search Google for structure skeleton, search for full structure. I'm going to have to assume some familiarity for this audience with INCHI keys. The first part of the INCHI key is going to get you to the connectivity, the skeleton for the chemical, and the second part is going to give you charge, isotope, layer, stereochemistry, etc., etc. So there could be a lot of variation in this second part of the INCHI key string if anybody happens to misrepresent the stereocenters, if they miss one, if they flip one, etc., etc. So we use INCHI keys all the time. So let's talk about how many chemicals we have relative to a lot of the other applications out there. So our data set is pretty small, right? We're approaching 900,000, ChemSpider's 90 million. Again, I was involved with ChemSpider initially. It was a hobby project. We built it in a basement for fun, programming from 10 o'clock at night till 2 in the morning, literally for fun. 2 years later, we sold it to the Royal Society of Chemistry, and myself and the small ChemSpider team went to work for RSC for a few years. I was there for 5.5 years. When we brought up ChemSpider initially, it was 10.5 million chemicals. The initial set was harvested from PubChem. We grew it to about, I don't know, 40, 50 million chemicals before I left. Now it's approaching 90 million. And it just keeps growing. They just keep adding more and more chemicals, which is great. PubChem is about 111 million, may have added more since I last checked, and CAS registry, I think they're adding 50,000 chemicals a day, I think that number's correct. It might be a week, but massive. Compare those other databases to public domain, ChemSpider, PubChem, CAS, commercial access, and ours 900,000. So we're tiny. This is an incredible effort to pull up all of these data and make them available. Massive resources, massive investment in computer systems, the amount of work to pull this together. And there's so much benefit of having all of these data available. We can't say enough thanks to what PubChem has done and what ChemSpider has done over the years. Not thanks to me, because I left. But there are hundreds of these websites at the moment, and there is also, I'll talk a little bit about the blood exposome, another great effort that is coming online, of course. So I gave a talk recently about what constitutes the exposome, and our application is being used for that. We're trying to characterize what's available, and I mentioned a little bit about the focused chemical lists. So our specific effort isn't just about chemicals. You can see how it breaks out into, you know, toxic substances control, drinking water, pesticides, etc. So those little lists with this 900,000 becomes very, very important. It's very useful because to have them segregated that way. Let's flip back now to this vomitoxin story. So if I search for vomitoxin based on INCHI-key, the first block on ChemSpider, I will find 19 of them. 3 of them are isotopically labeled, so I can ignore those because maybe they're specifically labeled for a purpose. Maybe it's for doing mass spec studies or some type of metabolism study. That leaves you 16 chemicals called vomitoxin that are likely all expected to be that chemical. They're not flipping stereo variants for a particular reason. This is a very complex chemical. They're not going to be synthesizing these probably. So we have potentially 16 chemicals that are all supposed to be the same chemical. That's a lot of dilution in a data set. If we now go to PubChem and do the same, there are 33 unique INCHI keys. Again, some of these are isotopically labeled for particular reasons. But the one that PubChem elevates to the highest level in terms of supposedly being vomitoxin, they have approaches to do that, turns out not to actually be vomitoxin. So the 33 unique INCHI keys here, 19 in ChemSpider. This is dilution, and this dilution causes a significant amount of problems when you're using these data for specific purposes. So these other databases are growing very quickly. We're only adding a few thousand chemicals between releases. So between the last 2 releases, we only added 6,000 chemicals, I believe. They can add 1 million chemicals overnight to these systems. So as an example, vomitoxin has 7 stereoforms that are all generated in a computer as part of the zinc effort, so that they can use these in things like docking exercises. So we are trying to get another 20,000 chemicals in the next 6 months. But again, it's going to be slow relative to these enormous databases because we're trying to not dilute our data sets with what I think of as distracting chemistry. As an example, this is an old snapshot of ChemSpider, which had 81 million chemical structures, and the largest deposition is from one chemical vendor. It is 52 million chemicals that was registered in September 2020. So 52 million chemicals from one vendor. I judge this to be a set of what I call MODs, make on demand chemicals. They likely have never existed, but this chemical company is saying they can make them. Nobody's holding 52 million bottles of chemicals to sell. If you go through the next four levels, it's 14 million, 12 million, 9 million. All of these top 6 are all vendors or people who are mapping vendor collections. We don't do this. We do not add chemical collections from vendors. And just to see how bad it gets, this is ChemSpider with Taxol, 79 different results for Taxol in Shiki. I've written a number of articles over the years on data quality in these web-based databases. Larger is not always better, and we have to do, I think, better work to make sure that we are removing some of the stuff that we don't need, so that we are not breaking the internet with low quality data. It's hard, it's challenging, especially when you're at 100 million chemicals. So there's a whole set of articles out there. By the way, we're still cleaning also. This is one of our most recent chemicals that I found. This is level 5. This made it out to the public domain. This is a chemical that was inherited from a PubChem registration. And I don't think any of us would really like that particular chemical as a structural representation. It's bad. So you can see level 5 is programmatically curated from either Actor or PubChem. We've got low confidence in it. But it did end up in our public collection. We have ways to try and curate this stuff automatically. This is an example on PubChem, by the way, which when other people are registering their own data in PubChem, there is so much terrible chemistry that gets pushed in. It's really bad. Oh, the difference is these are 2 representations of the same chemical. Ours is the inheritance of that one on the right-hand side. So our curation efforts require highly skilled people. We have written this up November of last year. There is an article, open access, if you're interested in how we built it. Going back to lists, this is our tire chrome rubber. When we write a report, we curate all the data, we make it online. This is our terpenes in vape. So that presentation out in Seattle before COVID hit. Actually, the week before COVID hit, I might have been on the flight that brought it to Raleigh. I had to put together a list of terpenes in vape. Hydraulic fracturing collection. So these lists are all there for you to use. The MS Ready structures is our effort. I believe Andrew McCachran is listening in on this call. He was the first author on this. This is our way to take, somebody might need to mute. This is our way of taking the chemical substances that we have as structures, and we de-stereo, we split the multi-components into individual structures, and we desalt and neutralize. Then we map all of these MS-ready forms of the structures across all of the substances that we have. So this allows you to have this mapping between these MS-ready forms and everything. The best way to look at this is in this particular image. This is generated with Emma Szymanski for a publication. She generated it. She did the hard work. So the MS-ready form of nicotine is DL nicotine in the middle, and you can see going around, we have the L form, the D form. This is the salt. So we desalt, de-stereo. They all map to this. This is the isotopically labeled, maps to this. This is the multi-component, maps to this. So searching on the formula or mass or Inchi key, first block, for this will link you automatically to these other forms. This is great work that Andrew McCachran did when he was with us. And for any chemical now where this has been done, you can come from the linked substances. You can see the MS-ready forms. Click on this and it will take you out to the 18 substances. So now you find all of the associated salts. And by the way, in the depiction, we filter out the isotopically labeled chemicals. We map them, we retain them, but we filter them out in the display. From here, from this set of chemicals, you might want to know what's the phys-chem property? What is the what hazard data are available? Are there bioactivity data? You would send it to batch search, and then you would harvest all of the data of interest, CAS numbers, names, DTXS IDs. So any multiple results page gets you to that. Let's go now, because we've done all of this, we can use the mass and formula searching. In the advanced search, you can come in and search on a particular mass. Every structure, every MS-ready structure has mass and formula, and then you can perform those searches. So this 191 plus or minus 5 ppm search is going to harvest you a list of chemicals. We've filtered out the multi-component chemicals because the individual mass could be for one component only. And then from here, we want to try and rank from this list. This is 329 chemicals. Which ones of these might be more amenable to detection in an environmental sample? And how would we do that? That would use some form of metadata ranking. And we'll talk a little bit about that. But this is just the mass searching right now. Formula searching, you can search based on MS-ready formula, that distillation that we've done from salts, multi-component chemicals, or the exact formula for the multi-component. So if I do an exact formula search for this particular formula, exact formula, so however many components are in there, we get 3 hits, and that's it. If, however, we do an MS-ready form, that means that any of these chemicals that could be available as a salt form or as part of a multi-component system should give us, in theory, a couple more hits, maybe. Well, it gave us a lot more hits. It gave us 122 more hits because EDTA, which is what these are, the variant stereoforms of EDTA, have all of the individual salt forms plus multi-component systems available. So that MS-ready formula search is significantly different. That's why we give you both. We're telling you what the individual components could be in a particular substance. From that 125 chemicals returned, 8 of them are single components, 3 of those 8 are isotopically labeled, and 3 of them are neutral, 2 are charged. But by doing that MS-RODI mapping, you collapse them all into a single search. So now we go from the singleton formula search, the singleton mass search, into the batch search. And I've shown you the benefits of batch search, right? What's the list of chemicals for a specific formula? What's the list of chemicals for a specific mass and error? Once I've done those searches, can I now pull those data into Excel format, SD files, and can I add additional information? And the answer is yes, absolutely. So in this case, we've come through monoisotopic mass. By the way, in the interface, these informational icons explain to you how these searches are done, reference relevant publications with DOI, and in this case, we're doing a search on these masses, each with plus or minus 5 ppm. And I would only, for mass searches, I would only recommend a couple of hundred masses because each mass can expand to hundreds of chemicals, of course. This is what the search file would look like in Excel. So this is now putting in a specific, of using formula instead of mass here. So you input a formula and it is matching multiple DTXS IDs using that MS-ready matching. Here's the CAS numbers that we include, the name, and then of course, all of that other information that you can simply go check through the batch interface. So this is one formula, this is the other formula, etc., etc. So now you get to look at this information, and the metadata that we have available for each of these chemicals is what starts to become useful in trying to make a call on the likelihood of detecting a particular chemical. And we'll go into that in more detail. I might also want to ask the question for a list of chemicals that I'm harvesting in Excel, not only what properties are available, but what lists are available, what chemicals are in what particular lists. So I can see here whether or not there are reference spectra available for any of the chemicals I'm searching based on CAS name, INCHI, mass formula, etc. So we can check these boxes to find out what chemicals are in. And in this example, going back now to that list of opioids that we had previously, here's my inputs, here's my list of DTXS IDs, and this is telling you where there are reference spectra. So codeine has reference spectra or details in each of these. So now you have an easy way of finding this across the different lists. The benefits of us bringing together all of these data through this batch search with hazard data, with bioactivity data, with physchem data, is that we can now pull all of this information together. And when we're using things like non-targeted analysis, we want to try and prioritize those chemicals that we want to examine in more detail. If chemicals are determined to be very high hazard, high toxicity, or we have come up with ways to use exposure modeling and the hazard data to give us a high risk, we want to focus our efforts on those primarily. So this is a way that we want to pull all of everything through the dashboard and then rank the candidates for attention. I mentioned chemicals in commerce, and this database is not available to you publicly in this form. This is Factotum. This is how we have been pulling the data together to push out to the dashboard. So you can see we've looked at almost 600,000 individual products on the market, 511,000 related documents, 3.9 million chemicals extracted from all of these product lists. So think of safety data sheets, databases of chemicals in commerce, etc. And this is mapped so far, it's mapped to 27,000 unique substances in our application. We have 882,000. To date, we've only been able to map 27,000 of those to chemicals in commerce. This shows you how it's all broken down, so you can actually go through here and you can see we've got arts and crafts, home maintenance, personal care, pesticides, and we can drill down even deeper. So if we want to ask what chemicals are in a shampoo, you can actually go into the interface on the home page, click on products and use categories, type in shampoo, and you will find a list of every chemical in anti-dandruff shampoo. You could find every chemical that's in a hair coloring, things like this. So for example, this is looking at methylparaben. Not only are we pulling the mapping between the chemical and the product itself, we can also give you weight fraction details for these chemicals in different from different sources. And this methylparaben has 462 products mapped to it. These are the actual product names. So going back to the mass spec stuff now we have the number of products. Now we have the toxicity data. Now we have the phys-chem data. We have how many lists contain that chemical. All of these become important metadata filters. So historically, when I was involved with ChemSpider, we wrote a publication on known unknowns utilizing mass spec data and ChemSpider. So some of you will know this from 2012. And what you're doing is you're using a formula search or a mass search to get a set of hits. I've shown you we can do that. We can harvest a list of hits from the application. And then somehow we need to rank and sort those. This, again, is a lot of work that Andrew McCachran did. Andrew's now on the West Coast. He's out there working with Agilent. So these are some of the sources that we looked at using. So the number of lists associated with the chemical in the application, PubChem data source counts, PubMed reference counts. This is always changing, of course, and we only have a snapshot of it. Whether or not we have bioactivity data, how many products it's involved with, and on and on. These metadata now get used. So when we do a search of this nature, we can switch this tile view of the hits. Click on the top right, and then it switches you into this view of the data. So this tells you CP DAT count, consumer product, how many consumer products is a chemical in, how many sources, how many lists, if you like, is it involved with, how many PubChem data sources. And based on the number of hits you get in these PubMed reference counts, PubChem, this gives you an indication of the potential to detect this chemical in the environment, in a particular sample, in a particular stream of data. It's not always going to be right, but these metadata values are become highly critical. And this is what that looks like. So we repeated the original ChemSpider work, and by we, I mean Andrew. He did this work. So what we did was we took the data that were originally used, the data set of all of the chemicals that was used for that work, pharmaceuticals, dyes, veterinary drugs, etc. And this is a ranking of where the chemicals would list based on the number of metadata sources. This is ChemSpider now, not the dashboard, but ChemSpider. So this is the original work done on ChemSpider. Now, in those days, ChemSpider was just over 25 million chemicals. Today, it's 81 million. So does that mean that bigger is better? The larger a database, maybe you're going to get better hits. Well, I've told you already that there's a big dilution effect. And the chances of you finding a chemical that is a make-on-demand chemical that is never even been made and is in a vial, but created for a catalog listing, is equivalent to being hit by Sputnik. So you're not going to find it. However, there are many other chemicals, of course, that you want to do. So you want to break out these data sets as best as you can to focus on chemicals that you would detect. Andrew repeated the work, and he used our dashboard to do that. We were only at 720K chemicals versus the 26 million. So it was only 3% of the size. So how would the dashboard do to be much smaller, but more highly focused, more highly curated than the ChemSpider database? And what was the comparison? This was the original data set. We used exactly the same data set for the work. When Andrew did this, for the same 162 chemicals, the dashboard outperformed based on both mass searching and formula searching. And the number in first position based on candidate metadata ranking, the dashboard positioned number one in more cases. And you can see the data here. This article is online and open access for you to access. There was one chemical that was very poorly ranked. This is a plot of the type of metadata that Andrew used. So he used our data sources, which is our number of lists, if you like, the PubChem data sources, the number of articles in PubMed. Again, that's a snapshot we harvested. The presence in the staff ident database, we had that registered. That's from Germany, and then the Chemi, Swedish Chemicals Agency. And the biggest contribution definitely comes from our data sources. I've been talking about structures a lot. I want to quickly mention our work on UVCB chemicals. So these are unknown or variable composition. This is the specific definition. These are complex reaction products. So we can't draw the chemical structures. We can draw constituents that are involved in these reaction products, but not the whole mixture. And then the biological materials, corn oil, nutmeg, things like this. Emma Szymanski out in Europe and many of her colleagues are working on these complex series because many of them are homologous series where you can actually see the distinct chain length growing by CH2 or CF2 in PFAS chemicals or branching, etc. So these are all related homologous series, very, very complex. We want to figure out a way to actually try and keep these substances in our application. ChemSpider deals only with structures. So if it doesn't have an INCHI key, you can't find it on ChemSpider. We're on the other side of that world because from the toxic substances control inventory, the public domain, public aspect of that inventory, the active non-confidential, we have it as a list. Over 14,000 of the chemicals out of 31,000, 14,000 of them are UVCBs. We don't have structures. They have CAS numbers. We can have hazard data. We could have lots of data. We don't have structures because they're UVCB chemicals. So 14,000 of them. And the way those show up are like this. So lard oil, hydrogenated tallow oil, palm kernel oil. We do have individual chemicals in Tosca, but we have lots of these UVCB chemicals. We have come up with ways to use Marcouche representations to represent these. So this is pretty clear. Xylene would be an ortho, meta, or para form. Xylenes could be a mixture of these. This is an alkyl benzene sulfonic acid with a chain length of C10 to C16. So these are Marcouche representations of the chemicals that we put online. And then we can enumerate these informatics representations to get a class of chemicals enumerated, or we can simply manually map members of this class. So here's an example. Linear alkyl benzene sulfonate. This representation here shows you that the methylene groups X and Y, we could have 1X, 2Y, 3X, 5Y, etc., etc. We have those mapped to individual chemicals that we have CAS numbers for, etc., etc. So this UVCB mapping is very important for us in the regulatory side for sure. We've also built in the abstract sifter. This is the mapping to PubMed. So we use mesh terms to look for the particular chemicals. They don't have to have structures. It's not using structure in any way. It's using CAS and name. And we can select specific queries to go against PubMed. So for limonene, I'm interested in any articles about food and exposure. This query comes straight out of the database. We populate it with the CAS number and the name. It loads 70 chemicals on the 70 articles on the fly. It's hitting 30 million abstracts in real time. This search would take about a second. It pulls down that list of 70 articles, It holds them in cache, and then we can filter through. So I can add in these sifting terms, limonene, food, exposure, and it will do a count, a frequency count of those terms, and highlight them directly in the abstract. There's also a version of this that runs in Excel. You don't need to use our application. And if you wanted to use the Excel version, you could have 100 chemicals, and it will go and harvest this information from PubChem in real time. This is the title, author's journal. And of course, if I want to read the actual article, I just click the PubMed ID and I go out to the PubMed directly. So this is the sifting terms. So the functionality is called Abstract Sifter. There's an open access article and a version for Excel. Work in progress, very quickly to walk you through some of the things we're doing. We're registering new lists. We never stop. I think I've added 5 lists in the past 6 days. We are working right now on a project where we need to flag categories against a certain set of 1,000 chemicals. We're looking at the USGS chemicals in water, the seized drugs, SWG group, synthetic cannabinoids. It's a personal interest, not as a user, just to be clear, but because of the amount of information we're getting about exposure to these in the waste streams. And then the blood exposome database, which has been a great effort. So thank you so much to the effort that's going on from Oliver and Dinesh to do this. We have downloaded the data set, and pulling these large data sets together is really challenging. It's great work. We're going to do some comparison with our abstract sifter approach and see if we can work together and compare notes on this. We already have a list approaching 20,000 chemicals from our curation efforts on the blood exposome. Of course, downloadable. We've also taken our MS-ready forms of the chemicals. Again, this is work I should acknowledge Andrew McCachran for being involved with. We took those MS-ready forms. We predicted the in silico fragmentation using David Wishart's work out of Alberta. So we predicted positive ion, negative ion 3 energy levels plus GCMS, EI, electron impact work, generated all of those data and put them in a database. So we can now search experimental spectra against predicted in silico spectra. So now we have in silico matching, plus we can use metadata to try and rank order. And this has been done very successfully. We have a new dashboard that's online called the Hazard Comparison Dashboard. That allows you to create a heat map of toxicity data for hundreds and hundreds of chemicals, pulling on the data that we have. We've structure, substructure search, and then our API and web services. I'll tell you a little bit more. So this is the CFMID work. This is all from David Wishart. We predicted all the data. We built our own viewer. So this is our prototype. It's not available publicly at the moment. But ultimately, this will be accessible through the dashboard. This allows you to input mass or formula and choose which type of spectral data you're generating, and then use the data from the spectrum to do the search. And then it pulls back a list of the chemical substances based on the match factor between the experimental and the predicted. And then you can view, of course, the chemical structure associated. This viewer allows you to hover over the individual fragmentation peaks and see what the fragments are. While the application is not available through the dashboard, all of the data have been published. They're available if you wish to take advantage of the fact that we generated all of these data. The publication is on Nature Scientific Data. All of the data are available for download. And we've also published on how successfully this has been used to look at some of the spectral data we got from our EPA non-targeted analysis collaborative trial. NTACT is EPA non-targeted analysis collaborative trial. It's an effort that's been going on for a number of years. This is our structure, substructure similarity search capability. Input a number of substructures, the formula, and it will utilize that information to identify chemicals in the dashboard. Couple that now with CFMID and candidate metadata ranking, and we're going to move things ahead even further. Andrew also published on the work he did to apply this approach against the CASME data sets that were available. So we've used the candidate ranking and the CASME work to look at 5 years of CASME data. Great performance, really, really happy with that. My present postdoc, Charlie Lowe, is working on amenability predictions, so we can figure out for a set of chemicals which ones you would expect to identify in positive ion, negative ion, GCMS, because some of the chemicals we're studying, of course, are silent in specific methods. So he's building those models. We've been harvesting data from many different sites, and this is going to be published shortly. Andrew was kind enough to provide us with some data he measured, and the model's looking very good. This is some of the visualization of those data. We also want to add more information to the database. So this is the work that's been done from PNNL. They've provided us with a collision cross-section database for many chemicals they've predicted. So we will be putting that in once time allows. We do have web services available. These are archaic, they're old, they're functional, but we are building a brand new API on the new application we're building right now. So you can use these web services if you're interested. It will give you access to some of our data. Our data sets are available for download. We continue to update them on an ongoing basis. Please ignore this date right here. It's the original posting, not the update. You can, of course, use all of the services that I'm mentioning for free that give you the data back in JSON and XML for you to use. If there are any data sets that I can provide you that you have interest in, let me know and I will do my best. And here's an example of using a particular service call using the INCHI key, and it will pull back the DTX CID, the image, the mapping directly to our chemicals, etc. These are being used by the community already. So we work with Norman, and they link their Norman data sets to our data sets on the dashboard. So they can link from their systems directly into the Untitled Chemicals Dashboard. And we do the same in the opposite direction. We go back to their data sets. We're trying to do the round robin tripping between systems. And this is where they've taken the data and used it in MetFrag. Again, our data is accessible to anybody for reuse, repurposing. This is the linkage between MassBank and directly using our DTX SID, so that you can come from the MassBank record in to see all of the toxicity data, PhysChem data that we have. This is taking advantage of what we're building directly integrated to another system. This was a lot of information to take in. It's like drinking from a fire hose. We're changing all the time. We're always building new systems, applications. There are a lot of resources available if you're interested. Again, this set of slides will go online to SlideShare, and I will circulate it to the group. But there are many presentations online, lots of videos that have been given over the past few years. There's a manual for you to access, and then the latest news as we go. This is my SlideShare account. So my name, Anthony Williams without the H. This is Figshare. Actually, you can find all of our posters, presentations. We did the last release in August. This is the 10th release of the dashboard. This will give you an overview of what happened with this 10th release. These are the primer videos. I think you say primer in America. I'm British, so I say primer. This will give you an overview, and this is the publication that tells the story about how we originally built it. So in conclusion, the access that we're trying to build is for curated, highly focused data of interest to us in the environmental toxicity, environmental screening, EPA focused world that we're in. Not trying to build the largest database of chemicals. That's not our focus. The MS-ready work that's been done, and we are building the system is almost finished. We will put online an MS-ready web service, so you can generate your own MS-ready forms by uploading thousands of smile strings if you wish. SD file is coming. So that we believe the MS-ready form is very useful for the identification of structures. The metadata facilitates ranking for sure. We've proven it many times. And then we're working closely to get these relationship mappings in place now with metabolites and degradants, etc. A lot of work going on there. I didn't show it. And then also this curation and sharing of data between ourselves and other systems such as Norman is useful. I have a request. When you use the site, which you almost certainly will find some issues occasionally, please tell us. It's a little bit like Wikipedia. You give us feedback and we will respond directly to you. We will tell you our feedback, whether we've made an edit on the database. This is our way of encouraging you to do crowdsourced curation. If you wish to have any lists of chemicals curated with any purpose for those, please contact me directly. And then I think these DTXS IDs allow people in links to information that you won't find in many other sites. This is a list of the names of many of the people I've been involved with to put this system together over the years. It's not exhaustive. They just come and go. And this is my contact information. I hope I've been able to give you a good overview of what we've been working on. And I look forward to any questions. Thank you.
Thank you so much, Tony. That was awesome and exhaustive and long. But hey, you know, congratulations. It's a massive amount of work, massive amount of like resources, and there are some questions that I would like to give to you over the time that you have talked. Maybe starting from Lloyd Sumner, can you tell how many MS and NMR data do you actually have in the database? And how much of those MS and NMR data would be experimental data compared to computational data?
So the NMR data, I would say, is very limited. There's a primary reason for that is that I work in RTP. We don't even have an NMR instrument here. I'm an NMR spectroscopist by training. If we did, believe me, I'd go down and enjoy shimming. No NMR instrument here. Mass spec, yes, we do a lot of LCMS, GCMS. The mappings that we have in place, I would imagine, probably accounts for about 8 to 10,000 of the chemicals that we have on the dashboard likely map out to public resources at this point. We have an ongoing project to curate the NIST database. And the internal version of that, I think, is around 60,000. There are lots.
What do you mean by?
So many of the chemical structures in NIST at the structure level are incorrect.
Okay.
So if you look at the structure of cholesterol, it's got one stereocenter in NIST because it was built so many years ago. They assume that you know the stereocenters for cholesterol. So we're trying to get the mappings between the exact structures based on CAS numbers and names to the NIST webbook and the NIST GCMS database. So it's about 50 or 60,000 internally.
All right. So we have more questions. How easy it is to download structures and information? Do you have different formats to pick from, like SDF or JSON?
So right now, if you wanted to download our entire database in an SDF file format or in an Excel format or a CSV format, you can go to our downloads page, click one button and download them. The SDFs are going to contain V3000 mol files, so your informatics system is going to need to support V3000. If you do a search and you get 5,000, 10,000 hits, click one button and you can select what format you want. We don't provide JSON formats for download. We feel that the majority of users are scientists, not people who are going to consume JSON directly, but we have services for people to consume that.
Okay. So for compounds that have bioactivity data, is it possible to filter those data by target tissues and organs?
You can filter based on the specific assay. The assay itself is going to be mapped to, in the detail of that assay, it's going to be mapped to specific targets. So it will tell you whether or not it was measured in liver, for example. You would have to go through and select the assays of interest, and then you're going to have to download the structure sets from that. Each of the assays itself is mapped to all of the chemicals that were studied in that assay. You can download each assay one at a time. If you want the complexity of all of the data, that with all of that detailed mapping, you would probably want to take the SQL file that is available for in vitro. That's the data for the bioactivity. Download that and put that in your own database so that you could write your own queries against how you want to do that. If somebody wants to follow up with me about how to do any of these, they can contact me by email and I will get online and walk them through how to do it.
Okay. Is there a way to query reported concentrations of chemicals in published studies or from the bioactivity experiments?
So for each of the bioactivity experiments, for each AC50 value that is extracted, you can get the full response, dose response curve for every single curve that's been generated. So a particular chemical could have been studied and have 10,000 dose response curves. You can download all of those data. If you're looking for concentration values that have been identified in individual studies, and that's not going to be particularly easy to do, you can go into the toxicity value table, which is under the hazard subtab, and there we will give you values such as regional screening limit values that have been extracted from some of those studies. But individual studies with individual identified levels, no.
Okay. The dashboard utilizes many identifiers. Can you convert these identifiers in the dashboard?
Yes. So if you have a list of INCHI keys, you can convert them to CAS numbers, names, DTXS IDs, smiles. If you have a list of synonyms, you could put in a list of 1,000 chemical names and get the CAS numbers. I would go through the batch search for all of that.
Mm-hmm. So I have one of my students is looking at CFMID currently, and looking at CFMID 4.0 and whatnot. So he's asking, is it possible to get early access to this tool, to your CFMID experimental comparison tool? He would like to touch on insights you found on what works best for drawing meaningful comparisons between experimental and CFMID spectra.
If we weren't under this very interesting lockdown that we're all suffering, I would invite him to come visit with us and hang out in Research Triangle Park for a week and embed him directly in the project. Unfortunately, everything is running behind our firewall.
Okay.
And
he's a starting student, so there might be a time when we don't have viruses around.
Exactly. Let's hope.
Okay, but we'll take you up on that. Actually.
Yeah, I'd love to do that.
So then we have a question: is there any database for gut microbes that you can recommend? I don't know where that comes from, but microbes and their chemistries.
Not off the top of my head, no.
All right, there's a Ines in Ireland, Ines Thiele. She runs a gut microbial database in
Ireland. In Ireland?
Ireland, yes, I think so.
Yeah.
Dublin.
So their guts are very different because of all of the Guinness. It's an entirely different world, believe me.
I love Ireland. All right.
In fact, I think my gut might follow. I drink a lot of Guinness. So.
Okay. Do other people have further questions? These are all questions I have read from the chat room. Awkward silence.
Is there going to be a little bit more of a demo? I thought there was a premise for that.
I can certainly try. As I commented, the issue we've been having is our network we're suffering dismally with network issues today. Okay. Actually, not just for today. So let's try. So
I was having a problem navigating. I was trying to work with you as you were going. But getting to all of the data seemed to be a little bit challenging unless you looked at it compound by compound.
Okay. So can you see my screen?
I can.
Okay. All right. So this is the public version. Everything I'm about to show you, you can do yourself. So let's just go for a list of chemicals for the moment, and I want to pick up.
All right. Pull down that I didn't see.
Yeah, so it's in list of chemicals, and I want to find, for example, tobacco. So here's a list of database of chemical compounds present in smokeless tobacco products. So this is from a database, I think, that comes out of UNC Chapel Hill. So there's 208 chemicals. So what I would like to do now is download this data set. So I can click here, download Excel, and now it's going to push an Excel file down to my desktop that includes the CAS number, the name, the smile string, the INCHI string, the INCHI key. And I can open that up. So this is an example of downloading the file. I don't have an SDF viewer on my desktop, on my laptop here. So if I downloaded an SD file, I wouldn't be able to open it directly, but I can go into remote desktop to show that if you're interested. So
Well, when I
here's,
go, can I back up here a little bit?
yep.
When I go to the dashboard, I don't see that same list at the top of the screen that you were pulling from. So I don't see home, advanced, batch, lists, predictions, or downloads.
Do control, just do control F5 to refresh, or F12 hard load cache reload, because that is the header of the application. So if you're not seeing that, I've never heard of that situation. I'm assuming you're using Chrome on a PC,
Yeah,
or?
yes, Chrome on a PC.
Huh.
Okay, I just tried to do, it's doing a refresh, but okay.
I can't think of why you wouldn't see the header. Is anybody, just in case, anybody else seeing that on the call?
I haven't tried it out myself. I wouldn't know.
So here's download if you want to download the SD file. So I'll continue, but there's no reason that you shouldn't see the header. I don't know why that would be. So here's what was downloaded. So there's the hyperlinked DTXS IDs. There's the preferred name, CAS numbers, INCHI keys, UPAC names, smiles, etc. So that's by default from the download page. If I have these 208 chemicals and I want to send them to the batch search, click one button, and now here's the list of identifiers that I've moved over, and then I can now go to download the CAS numbers, the INCHI keys, the UPAC names, etc. So that's, and then you can access any of the information that's available on a batch search. If I go back now to those list of chemicals and I take a list of CAS numbers, so Oliver was asking about converting between them. So if I go to this page and I'm looking for CAS numbers, of course I can now convert. I can simply download the name, the INCHI key, the UPAC name, the smiles, the INCHI string. So I'm searching on CAS and I'm pulling down all of the alternative mapped identifiers.
But these were only these 820,000 chemicals that you have, not like in ChemSpider.
Yes, that's correct. And the reason, and I don't think they have a batch search in ChemSpider like this.
No, I don't think so.
But I would be so one of the challenges that we have, as I said, is if you searched here, if you searched vomitoxin, you're going to get one hit. If you search ChemSpider, you'll get 19. Yep. Now you have to choose. Yep. So it's that difference. We will give you less hits, but higher quality. It's, you can't tune both of those knobs simultaneously, unfortunately.
Very good. All right. Other questions from other people here? I mean, we are like longer than usual. It's almost 11:30 on the West Coast. All right. So,
maybe I was mistaken, but I thought I had 90 minutes, and if I was mistaken, I apologize.
No. We schedule 10 to 11:30.
Okay.
To allow for questions. Yeah. You know, we do that, but you know, that's fine, because often enough you get like 10 minutes talking about your things, and you're like, okay, 10 minutes is hard.
So I didn't mention predictions, but if any of you have chemicals that we don't have on the dashboard, then you can use this ability to predict in real time. So you have all of these ecological properties and physical chemical properties that you can
generate. You predict on chemical damage or on microbial conversions? On what do you predict?
No. So these are all going to be based on extracted data for things like LC50 values from fathead minnow,
toxicity and log P and whatnot. Yeah.
Yes, exactly. But I have a feeling that you see this spinner right here. I think we have a problem with our network is struggling for 2 days.
Well, you know, presidential debate. Why
That's causes a major distraction. You're not kidding.
Why should government databases be any better? Anyway, so thank you so much for your insights here, and I really wanted to bring it out to our community. I have seen you several times now, in the last, I think, 4 weeks. So thank you again. But that's why I wanted to invite you, like that we see it also for our community, and that's what we have to do. We have to give it to the communities.
Yep.
So thank you so much again. Next.